So, good afternoon, all. Today, we will be starting with the lecture on the stem cell. At the end, we would be able to define what are stem cells, would be able to differentiate different type of stem cells and where do they come from, help know the methods of regulating different activities of the stem cells, and how these stem cells are used in regenerative medicine. What are the stem cells? These are unspecialized cells capable of dividing and renewing themselves and capable of differentiating into specialized cells. So when the stem cell give rise to specialized cells, for example, the nerve cells, the blood cell, and the muscle cell, the process is now called as differentiation. Now, moving on to the next slide. Some cells divide and stay as stem cells, while others differentiate and cannot be stem cells anymore. Because in stem cells, there is a continuous expression of certain transcription factors, like the genes which we have mentioned, OCT4, NANONG, X, OX2, will prevent differentiation of the cell, and thus the cell can maintain its pluripotency characteristics. So what is the meaning of pluripotency? Any cell that is capable of giving rise to different type of cell lines. For example, you have already studied that the inner cell mass during the blastocyst stage. This is the one that will give rise to all the three germ layers, ectoderm, endoderm, and mesoderm. So they are pluripotent, right? Now, how can we identify the stem cells? They can be identified by labeling of several transcription factors which are found only in the cytoplasm of the stem cells. I could see in the diagram there where they have been labeled by surface proteins which are specific for stem cells only. On to this slide. What are the possible end results of a stem cell division? They can either divide into identical daughter cells, which are called as symmetric division, or they can divide into two different types of daughter cell, one being the stem cell and another being the progenitor cell, called as asymmetric division. So this progenitor cell is what is a proliferative cell that is capable of differentiating, but it has no self-renewal ability. Right? You can see in the diagram, this progenitor cell can give rise to different variety of cell lines. Depending on their potency, that is how they are able to differentiate into specialized cell type. They have been classified into various types. Totipotent stem cells, which is the zygote itself, which can differentiate into 220 different type of cell types found in an embryo, as well as it is capable of forming the placenta. So that is totipotent. The second is the pluripotent, which just in a few slides back I mentioned to you about those. These are embryonic stem cells now called from the inner cell mass and can differentiate into all the cell types of the body but not the placenta. Multipotent stem cells from the ectoderm, mesoderm, endoderm, and like in the adult tissue, they can differentiate into limited number of cell type and can replace damaged cells in the adult tissue. And the unipotent stem cells, for example, given into single cells, for example, in the skin and the liver, but not certain if they only differentiate into one type of the cells or not. So this is how we differentiate each type of the cell in our body as totipotent, pluripotent, multipotent, and unipotent. How from a totipotent, which is a zygote, it can give rise to pluripotent. The pluripotent can give rise to multipotent, which are the adult cells, ectoderm, endoderm, and mesoderm, and each of them giving rise to single cell type as in the unipotent. Embryonic stem cells are the stem cells which are derived from the inner cell mass of the blastocyst of the embryos before the implantation in the uterus. You all know the stage of development. So how can we obtain large number of embryonic stem cells? So when the inner cell masses are removed from their normal embryonic environment and cultured under appropriate conditions, the inner cell mass cells can continue to divide indefinitely and produce cells that have a potential to form any cell type. So this is how we retrieve the embryonic stem cells. As you can see here in the diagram, that they can be retrieved. How to direct the embryonic stem cells to become a certain tissue? They are cultured with a mix of growth factors and certain regulatory factors in order to direct these cells towards specific line of differentiation. For example, if bone morphogenic protein molecule it will induce the formation of the mesoderm. If it is activin or nodal molecule, it will induce the formation of the endoderm, and so forth. Like fibroblast growth factor will induce the formation of the ectoderm. So this way we can give rise, these cells are directed to form different type of cell lines. So what is their use? It has been seen that they are used in type 2 alveolar lung disease. If you remember, type 2 alveolar cells are the type 2 pneumocytes that secrete surfactant. And surfactant are very important to keep the alveoli in a inflated state, right? And that prevents neonatal lung disease. So it can be used in a congenital neonatal lung disease. Insulin secreting cells have been cultured and can treat diabetes mellitus. Despite of the great therapeutic promise of embryonic stem cells in many diseases, a lot of ethical and political opposition have been present with their usage. For example, main opposition is in harvesting these embryonic, which involves destruction of the human embryo. So ethical issues are involved, and also there is an opposition from the political class to the scientific community towards this part of the research. So the next is the adult stem cells, which are undifferentiated cells in a tissue or an organ, and that can be differentiated to give rise to some or all of the specialized cell types of that tissue or an organ. So the primary function of these adult stem cells is to maintain and repair the tissue in which they are found. They can also remain non-dividing until they are activated by the need for the new cells to maintain the tissue or due to tissue injury. The adult stem cells have been identified as bone marrow, blood cells, skeletal muscle, skin, gut, liver, ovarian epithelium, and testes. So how can you regulate the number of the differentiation of these adult stem cells? These adult stem cells are located in a specific areas of each tissue, which is called as stem cell niche, and the microenvironment of each niche will control the stem cells. So what is this stem cell niche? It's an area of the tissue that provides a specific microenvironment in which the stem cells are present in an undifferentiated and self-renewable state. Cells of the stem cell niche interact with the stem cell to maintain them or to promote their differentiation. Is it clear? So, the cells of the stem cell niche will interact with the stem cell to maintain them or to promote their differentiation. Different cells which are located in the niche are like, for example, the endothelium, the nerve cells, the macrophages, which have effect on the stem cell and will define the activity and the fate of the stem cells. So how the stem cells and the different type of the cells in the niche will communicate with each other? They can communicate directly to the cell-to-cell contact or indirectly through the secreted factors, the promoting the growth factors. What would happen if we change the niche around the stem cells? They may change their differentiation. For example, the human neuronal stem cells can produce muscle cells when they are implanted in the skeletal muscle. Interesting, is it not? So, how the patient benefited from the adult stem cell research? Yes, over millions of patients worldwide have been treated with adult stem cells and experienced improved health in diseases such as diabetes type 1, which is juvenile type, liver cirrhosis, osteoporosis, acute heart damage. Disadvantage is that it is restricted in their differentiation, not pluripotent. That means it cannot give rise to all the tissue types. They have a short lifespan in vitro. They cannot proliferate much in the culture. And so the research continued for another source of pluripotent stem cells. And that brought us to a new variety of stem cells, which is called as induced pluripotent adult stem cell, which we'll see in the next slide. As I said to you, with the fallacies that were present with the adult stem cell, now we have a new one which is called as induced pluripotent adult stem cells. So yes, it is possible because now we can know that certain genes become inactive in these mature differentiated cells, and we know that if we can produce or activate these silent genes, then we will produce certain transcription factors that will reprogram the cell to become a stem cell. So the first person to do this was a Nobel laureate, Shinya Yamanaka of Japan, who proved that activation of 4 specific genes, which are mentioned MYC, OCT, SOS, and KLF4, will produce specific transcription factors that would reprogram the mature, for example, here the skin fibroblast to become a pluripotent stem cell. These stem cells are called as induced pluripotent stem cells. So what are these? These are adult stem cells that have been genetically reprogrammed to an embryonic stem cell-like state by being forced to express genes which were silent and factors important for maintaining and defining the properties of the embryonic stem cells. So what are the induced pluripotent stem cells capable of? Yes, they can show that they show all the stem cell markers. They are capable of generating cells from all the 3 germ layers, ectoderm, endoderm, and mesoderm. And using this technology, they have reprogrammed scale cells into more active motor neurons, eggs, sperm precursor, liver cells, bone precursors, and blood cells. So see how interesting from where we started and where we have reached. They say, as you all know, that necessity is invention of it helps to invent many things. There it is. We had some ethical issues and some opposition for the adult stem cell. We came out with this new technology, and this is doing wonders. So why do we need to study and isolate stem cell? Yes, because in regenerative medicine they have been helping to repair the damaged tissue. Promising effects were seen with the treatment of diseases like Alzheimer's, Parkinson's, and what is this regenerative medicine? It's defined as a process of replacing or regenerating human cells, tissue, or organ to restore or establish normal function. For example, using stem cells to repair some of the damaged tissues such as bone, cartilage, muscle cells, bladder, of that matter, skin. So it's an ongoing process of research in stem cell. Many more advancement you will see in the future. I hope you understood the lecture. Thank you so much.